हेलो गाइस, आई एम शुभम फ्रॉम शो ट्यूटोरियल्स। इस वीडियो में हम डीबीटी के PYQ सॉल्व करेंगे। दिस इज द पार्ट 8 ऑफ दिस पर्टिकुलर सीरीज। ओके, so before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. This is absolutely free from your side, but it will help us grow. The first question: match the animal cell lines in list 1 with their commercial applications in list 2. So choose the correct answer. ओके, इसमें बी जो है माइलोमा सेल लाइंस, दैट इज वन यूज फॉर मोनोक्लोनल एंटीबॉडीज, राइट, एंड दिस इज प्रेजेंट इन ऑप्शन 1 ओनली. ओके, so this will be the correct. Then see the next options: simian kidney epithelial cells, polio vaccine. Okay, then porcine kidney cells, urokinase, D2, and baby hamster kidney cells, foot and mouth vaccine. Okay, so you have to remember the other things also, but हमारा motto ये है how you can solve quickly. Which of the following enzymes adds an extra adenine at the 3' end of a DNA strand? Extra adenine को add करता है at the 3' end of a DNA strand. Which enzyme? So that is tag polymerase. Okay, this is very important. You have to remember. Enzyme that adds adenine at the 3' end is tag polymerase. Myasthenia gravis is primarily caused due to myasthenia gravis. Basically, it is a autoimmune disorder. Okay, autoimmune disorder है caused due to defects in the cholinergic receptor system. Option C. Okay, which one of the following term is used to describe the protection of the unimmunized individual based on immunization of sufficient number of other members of the population? मतलब पॉपुलेशन में मोस्ट सफिशिएंट नंबर का अगर हमने इम्यूनाइजेशन किया, देन द अदर जो अनइम्यूनाइज है उनको भी प्रोटेक्शन मिलता है. तो व्हाट इज दैट कॉल्ड? एक्टिव इम्यूनिटी? नो. पैसिव इम्यूनिटी? नो. हर्ड इम्यूनिटी? यस. ओके, एंड पोस्ट एक्सपोजर इम्यूनिटी? इसका आंसर है हर्ड इम्यूनिटी. कोविड टाइम्स में ये हमने देखा है, मैक्सिमम लोगों का, अगर मैक्सिमम पीपल्स if they are vaccinated, then other non-vaccinated get the protection, right? Get the protection, and that is herd immunity. An integral plasma membrane protein is glycosylated. ये glycosylation का क्वेश्चन है. Scientists can trace the journey of this protein from the time of its initial translation to its translocation to the plasma membrane in its immature form. Which of the following changes in its molecular weight can be expected to take place during its maturation? Now see, प्रोटीन है, इसको हम क्या कर रहे हैं? ट्रेस कर रहे हैं. ओके, ट्रेस इट्स पाथवे फ्रॉम ट्रांसलोकेशन, ट्रांसलेशन टू ट्रांसलोकेशन, पूरा ट्रेस कर रहे हैं आप. एंड दिस इज अ ग्लाइकोसाइलेटेड प्रोटीन. So which are the changes in the molecular weight, okay, during its maturation? First option is gradual decrease after translation, मतलब molecular weight translation के बाद decrease होगा. Second option initial decrease followed by an increase. Third option, initial increase followed by decrease. Fourth option, there will be no change in the molecular weight. So I will tell you glycosylation में N linked है and O linked है. N link take place in the ER. O link take place in the Golgi. Okay, ER में पहले होता है, फिर Golgi में modification होता है, right? So it can be said that Golgi में modification होता है, तो कुछ ऐसे sugar molecules हैं that are removed, right? So the molecular weight will decrease first, and then complex sugars are added. Golgi में complex sugars add होते हैं. स्टे ट्यून, हमारा सेल बायोलॉजी का प्लेलिस्ट चल रहा है, उसमें हम प्रोटीन ग्लाइकोसाइलेशन डिस्कस करेंगे इन डिटेल. ओके, तो दिस इज जस्ट अ बेसिक वीडियो. हम क्वेश्चन कैसे सॉल्व कर सकते हैं. तो ईआर में एन लिंक हुआ एंड गोल्गी में मॉडिफिकेशन होने के बाद कॉम्प्लेक्स शुगर्स ऐड होते हैं. देन द इनिशियली मॉलिक्यूलर वेट विल डिक्रीज and then it will increase after addition of complex sugars. Okay, so option 2 will be correct. Which of the following fragments, regions of an antibody molecule, will you use to identify the presence of specific epitope in a biological sample? Okay, specific epitope हमें identify करना है. And what you will use? We know this epitope is basically antigen which will bind to antibody. Antibody में binding site है paratope. So antigen will bind to this paratope of antibody, right? Then antibody का कौन सा fragment हमें use करना है? Variable fragment FV, a variable region which will recognize the specific epitope. Got it? Again, match the column है least 1 with least 2. This is also easy. जो B है ribonucleoprotein complex to bind to the signal peptide. ओके, मतलब राइबोन्यूक्लियर प्रोटीन का कॉम्प्लेक्स है जो सिग्नल पेप्टाइड को बाइंड करते हैं. दिस इज बेसिकली 1 SRP, सिग्नल रिकॉग्निशन पार्टिकल, okay, which bind to the signal peptide and it is a ribonucleoprotein complex. So B1, again this is present in option 1 only, option number 3. Okay, then जो दूसरा है C, formation of correct disulfide bonds in protein. Disulfide bond का formation PDI, protein disulfide isomerase C2. Chaperone in the ER lumen is 3 BIP. Okay, D3 and A4. Lipid anchor for proteins attached with the membrane. Basically this is PPI A4. Okay, single option से ही हमें मिल जाता है, right? In the manner following. Given below are two statements. One is labeled as assertion A, other is labeled as reason R. Termination of DNA synthesis occurs upon incorporation of nucleotide lacking hydroxyl group on the 3' carbon of the pentose sugar into the growing DNA chain. मतलब जो OH group है वो नहीं चाहिए to terminate, right? The hydroxyl group reason, the hydroxyl group on the 3' carbon of the pentose sugar is involved in phosphodiester bond formation. So yes, both are correct, right? In the light of above statements, choose the correct answer from the option given below. Both A and R are true, and R is the correct explanation of A. So option 1 is correct. Right? We know chain termination. सीक्वेंसिंग में हम यूज करते हैं, राइट. डाइडिऑक्सी न्यूक्लियोटाइड ट्राइफॉस्फेट वी यूज टू टर्मिनेट. बेसिकली 3 प्राइम OH नहीं होता इसमें, राइट. नो 3H. एंड दिस इफ हमें पता है 3 प्राइम OH का रोल क्या है, फॉस्फोडाइस्टर बॉन्ड का फॉर्मेशन करना. तो इफ द 3 prime OH is not present, then no bond formation will take place and chain terminate हो गया यहाँ पे. So yes, this is correct. Which stage of mitosis is blocked by colchicine? Metaphase, option 4. Okay, colchicine is a drug, right? It inhibits or blocks metaphase stage of mitosis. You can see जो match the following है, those are very easy to solve, right? Now यहाँ पे G0 phase है, G0 phase. क्या होगा? क्वेश्चन phase, right? D1. So D1 is present in option 2 and option 4, right? Now let's see C. C option 2 में 3 है and 4 है. Now let's see. C M phase. 4 क्या है? 2 daughter cells are produced. So yes, this is correct. Option 4 correct है. Okay, C4, D1, B3, G2 phase है. This is new proteins are synthesized and cell approximately doubles in size. Growth phase है, इसमें सेल की साइज डबल हो जाती है, as well as new proteins are synthesized. And जो A है, that is 2, S phase of cell division, DNA of parent cell is replicated, right? So option 4 is correct. Okay, so हमारे 2023 के PYQs पूरे होते हैं in this video, right? And approximately we have solved 80 to 90 questions. Okay, 80 to 90 क्वेश्चन हमने सॉल्व किए हैं. And if we do the analysis, अगर आप अटेम्प्ट करते भी इसे, then 3 मार्क्स का एक क्वेश्चन होता है. ओके, विदाउट नेगेटिव मार्किंग यू कैन स्कोर 240 मार्क्स, राइट. 240 मार्क्स यू कैन स्कोर, जितने भी हमने किए हैं. So now I will show you the cutoff basically of that particular year. Okay, 2023 DBT JRF biotechnology eligibility test, right? So you can see 241 मार्क्स का हमने सॉल्व किया, right, without negative marking if I tell you. And last cutoff is 221 for the JRF. Category 1 is basically JRF. So more than enough है to qualify. Right. And as you can see for your particular category also. Some questions like match the following or statement based questions which are very easy. राइट, टू सॉल्व। ऑप्शन से ही कुछ क्वेश्चन सॉल्व होते हैं। ओके, and if you practice, if you do the syllabus, and you can achieve a good score as well. So that's it for this video. I hope that this video was helpful. You can find the playlist, ongoing playlist, completed playlist in the description, and I will recommend that please watch the series in the playlist only. ये मतलब सीरीज में आपको समझेगा. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel, and stay tuned for the upcoming videos. Thank you.